Welcome to the Adora introduction workshop on the R2 platform. My name is Jan Koster, and I'm heading a group of bioinformaticians in the LEXOR department who does all kinds of omics types analysis, mostly in cancer at this moment. And in this process, we've also developed a web-based platform called the R2 platform, which we basically support on the internet and where people can actually make use of this platform in their daily work. And today what I'm going to do is I'm going to show you a little bit how is this program working, what's the concept behind it, and with a number of demonstration slides I will show you how you can use it, how you can test hypotheses and these kind of things. Hoping that this will be helpful for you guys as well, and that you get the vibe of it and want to use it for your own work. So I've basically got around 1.5 to 2 hours of talking on what kind of things you can do in the platform. I will start with an introduction of how to get started, what is it, show you a little bit how you actually work in the platform, and then in the second hour I will go a little bit more into intermediate difficulty functions, and I'll show you a couple of examples of what is also possible without explicitly completely giving away how you do these kind of things. And then afterwards we have 2 hours planned where you can use your own laptop and see if you find it interesting enough to also try this on your own using the tutorials that we have, documentation that we have, or maybe you already have questions that you would like to try and answer. And in that session there also is going to be help if you need it by the R2 team that is also walking around here. And well, let's just get started. Wonderful, guys. So the R2 platform is a genomics analysis and visualization platform, as we like to call it. And it's basically consists out of two things. We have a quite extensive data warehouse where lots of data, mostly from public domain, but also private data sets, are already preloaded in our data warehouse. And on top of that, we have a web-based suite of tools that allows you to actually make use of these data that we have in the database. And the kind of data that we have in the platform spans basically all types of omics that you can imagine. So we've got DNA data, RNA data. You can work with proteome, with metabolites, drugs, metadata, even some lipidomics data is in the platform. So there's really a quite vast array of possibilities that you can make use of. What I will mostly focus on today is transcriptome data, because that's the most easy to work with, and that's also used the most, so that it has a link with most of the people that are in the room here. So the R2 platform, just a couple of statistics. It has been around since end of 2005 when we started developing it. And over the course of time it has now been cited in PubMed for nearly 3,000 times. We've got over 500,000 bulk RNA samples already available for you that you can just use. These are public domain data. A couple of million single cells, and well, around 2,700 data sets that you actually can already explore when you are online. Yes. So today the demonstrations that I'm going to provide you with are mostly on either colorectal cancer as an entity or a pediatric cancer called neuroblastoma. And for those two diseases, there's not too much that you really need to know to follow this. Just a couple of concepts which I have put here on the slides. So with neuroblastoma, it's a pediatric cancer. Typically happens in young children, and there the prognosis is actually defined by a staging system, which we will have a look at in a couple of slides. And the most important thing that you need to know for today is that there is a particular gene, the neuroblastoma MYC gene and MYC, which can be amplified in these patients, and once that happens, that really has a very poor prognosis on these patients. So we will use this concept to show you a couple of examples. On the other side we have colorectal cancer. Colorectal cancer is of course also a disease that many of you know. that can be classified into different molecular subgroups. This is called the consensus molecular subgroup system, the CMS. And here we will mostly zoom into one type of these CMSs, which is CMS4, which is the mesenchymal type, which also has a poor prognosis. So the slides progress really slowly. So if you are on the R2 website, the web page that you will see in front of you looks like what you see now on the screen. It basically has a central setup with a number system, 1, 2, 3, 4. And what we try to do there is guide you towards what kind of a question you would like to get answered. One meaning that you want to look at a single resource or single data set or multiple ones. Today mostly we'll focus on the single data sets. The second part allows you to select a particular resource, a data set. The third, a type of analysis that you would like to perform. And then the fourth one progresses to the next step. So if you want to select data sets in R2, you simply click on it. And then what opens up is a table where you can select from all the resources that are currently loaded within the R2 platform. So you can see how many records are already there in your personal account. And you can use the indication, as you can see on the top in these headers, to make selections on types of cancers, number of samples, authors, maybe study number that you actually know. And that way you can select particular data sets. Once you've selected a particular data set, then you can just simply click on it to confirm, and then it will use that data set. Underneath every selection that you make, there will be this little record that you see in the bottom part, which shows you a couple of statistics of where did this data set come from, what was the intent of it, what kind of tracks are in it. Tracks are grouping variables in R2, and where we got the data from. Also, if you start using R2 and you find out that there's maybe a particular analysis that has been published that is not yet in the R2 platform, but you would like to make use of it, you simply can just drop us a line via email, and we will try to make sure that we also incorporate this dataset into the R2 platform. So basically, once you have selected your dataset, in this case we're going to use this colorectal cancer dataset from NUNJ, which has a little over 1,000 colorectal cancer samples. And on that we can now use any kind of a tool that we would like to have a look at. The most simple thing that you can do in R2 is just view a gene, which does exactly what it tries to describe. So essentially what we can do here is we can just select a particular gene that we would like to have a look at. Like I said, CMS4 is going to be the thing that we're going to look at mostly today. And this is associated with mesenchymalness, and vimentin is one of the genes that has a lot to do with this epithelial to mesenchymal transition part. As you could have seen in this drop-down that opened, there are a lot of annotations that are already preloaded with most of the data sets. So this one is a very extensive one. It has dozens of different features that are already available to you. These are grouping variables that you can make use of in your analysis as well. So if you have a look at a particular gene, then the typical view that you will get initially is the so-called YY plot, as we call it. And here what essentially happened is you have these 1,000 samples. You start with the tumor with the lowest expression of your gene of interest on the left side, and it just progresses towards the high expression on the right-hand side. If you hover over these elements, it will show you the metadata that's associated with it. The reason why we like this particular type of plot is that we can use the space underneath these plots to track the annotations as color-coded features. Also in the platform you can make these things appear and disappear at will, and in that way you can see or visually already appreciate that with this expression of this gene called vimentin, you can see in the bottom track, the one that has this purplish colors in the left towards the greenish colors on the right, that this CMS tumor subgrouping information appears to have a relationship with this vimentin gene. Another thing that you can then do is if you progress downwards on this web page, you can then also change the view of this particular visualization. So what we're going to do here is we're going to look at it from the perspective of a track, of a grouping variable versus the expression of this gene. We're going to make the plot look a little bit different. So we're going to use a violin plot. And in that way, we can then just simply click the next button. And now this visualization that we had, this YY plot, became a plot that's called a violin plot. And also here, at will, you basically, by using this gearbox that you see on the top, if you open this up, you then can change a lot of parameters, a lot of features of your plot, and thereby make it look as you intended it. You can use the information also in layers in these plots. So what I'm doing here is that I actually take the data points, color them by a particular feature, in this case the CMS classification, and what then also becomes a possibility is that on the right hand side where you see this legend, these things become interactive as well. So you can switch on and off subtypes of genes, of samples. And depending on the track that you're using, this can become a really complex thing, but it also allows you to test hypotheses that you may start to develop when you're working with the R2 platform. You can not only color these samples by grouping variables, but also by expression of any gene that is available within such a resource. So as an additional layer, you can use the gene expression of another gene to show what the pattern would be in these different CMS stages. And thereby again, you can get some additional information. You can use transformations, you can use color schemes, whatever is of interest to you. And you can also export these plots by using the Save tab on the top, where you can export it as either a PNG file, as a clipboard, or as a vector image to be used then in other applications. So depending on the switch of the type of analysis that you're using, you can either look at it from the single gene perspective, as I did before, and then have all kinds of visualization that's associated with that. You can also just take two different genes and associate those together on like a head-to-head comparison, which is just showing you this XY plots. You can use this in groups, as you can see in the gene versus track, but it's also possible to use categorical data against categorical data, and then it becomes the visualization as you can see on the right hand side. And all of these can also be switched in different visualization properties, where you can actually just learn a little bit more about these associations. The one on the right hand side is also something that we call the visual Fisher-Exact plot, which is essentially a nice way of representing these like group versus group comparisons, where typically you only have a p-value, but now you can also use this as a picture, and as an additional layer you can use color as another variable to maybe see associations that are occurring within it. So if you have a particular gene that you are studying, like in this case fermentin, you can also wonder, is there any annotation feature in the database that associates with my gene of interest? And for that we have a function that's called the klinischnitt. And klinischnitt you can find on the right hand side in this little menu box over there. And if you then click on that particular gene, what R2 will do is it will use the expression of, in this case, fermentin, and associate it with any annotation feature that's already loaded in the database. And if you start using R2 yourself and you create your own variables, these will automatically be taken into account as well. So you can also add your own features and tracks to it. And all of these can just be simply visualized then by using this magnifying glass, where you can then see the association between vimentin and whatever is found in the most significant fashion. So this allows you to quickly scan through a resource and identify something that fits with your gene of interest. So that was kind of what I would like to show you as a beginning on using a single gene. What we will have a look at right now is doing a differential expression analysis. And also there, the concept is really easy. You just use this option box number 3. You identify differential expression between two groups. And then essentially what you can do is you can start putting together the analysis that you would like to perform. So here we're going to use this CMS classification again. We're going to use a lemma test to run the differential expression analysis. If R2 has count data for example, RNA-seq experiments, then also automatically algorithms like DESEC become a possibility, such that you can then do the analysis in count space. If that's not available, then it will just regress to either t-testing or lemma or whatever is possible on those types of data. That really depends on the kind of data set that you're using or the data that you're providing to us. So once you've done this track, so the grouping variable that you would like to use, then on the top you will see the group 1 and group 2, where you define which parameter you would like to use. So in this case we're going to use CMS2 against CMS4 to identify genes differentially expressed between these two different subgroups. And then there are many parameters that you can set on these analyses. So typically whatever is already provided by default is pretty much okay and fine, but you can also change things like the false discovery rates. You can change the number of genes that need to be expressed. You can use all genes or just an intersection like kinases or something like that. So there are many different options that you can then tweak and really provide the data for your analysis. So once then the analysis has been performed, R2 will show you the result, which is here the result of the differential expression analysis. And what you can see over here is that it has identified quite a number of genes that are differentially expressed between these two subtypes. And they are sorted on the p-value, which you can also adapt if you want to by just using this header columns where it will sort in another way. Also here, if you have seen an analysis, if you have identified a gene that you find of interest, you actually can click on the magnifying glass, and then it will go back into this visualization that we just had, this detail view, where you can now see what the differential expression is really looking like on a sample by sample basis. What you can then do, once you are on such a result page, you can make all kinds of adaptations to this as well, just like I showed you in the previous slide. We can, for example, remove the subset variable that we just used. Right? We only looked at CMS2 versus CMS4, but of course there are other CMSs in there as well, so we can actually release that if you want to. We can also then reload it, and now with the same gene, essentially you can now see the whole data set. So now you can also appreciate that it's actually CMS4 that has more expression than the other ones, versus the other possible hypothesis that it would be the other way around. Also here, you can make all kinds of adaptations to it, and then make it visually appear as you find appealing. And then there are these green bars that you can see on the top, which is essentially the result that came from the differential expression analysis. So what this allows you to do is that you can also now browse through your differential expression analysis using this adapted view that we made. So we now have included the other CMS classes, we've made the colors a little bit different, we've changed the dot sizes and things like that. And now we can just progress through this differential expression result using these boxes that are on the top. So now we can just go from one gene to the other, and thereby appreciate that you actually can see all of the times that this CMS4 group is actually sticking out in a positive sense. So another kind of analysis that you can perform from within the platform is you can associate also survival information. So if for a particular data set this information is provided, then essentially what you can do is you can now see that here we have this gene TGF-beta3, which on the right hand side we have this box called a Kaplan scanner. If you click on it, it will now use the information of that particular gene and see whether expression of this gene has the tendency or can be used to separate the cohort in a group that has a better survival versus one that has a worse survival. And essentially the test that we are doing is quite a simple one. We just cut this data, or we sort the patients again from low expression to high expression of your gene of interest, And then we cut the resource in two sides. And we start from the left-hand side, so a small group versus a big group. And for every time, we just move up one sample and then redo the test. And while doing that, we keep track of what the statistical significance is of that particular test. And this is a thing that we call the Kaplan scanner, and it allows you to identify an optimal separation in your cohort for segregating this cohort on the basis of your gene of interest. It also comes at a price because we're actually doing repeated testing on the same resource. So if you start using this kind of feature, you always need to provide the Bonferroni corrected p-value, which then also takes into account the fact that you did multiple testing. It allows you to pick up things like known associations that are like at 30% of a cohort or like 25% of a cohort, instead of using the commonly used median of a cohort, right? So there's no natural sense why you would always need to subdivide your cohort in two different groups if you know that a particular feature that it associates with only happens in 10% of your cohort, right? So these kind of things are automatically homed into if you use an algorithm like this. But in addition to this Kaplan scanner that we have implemented in the platform, you can also make use of medians, quartiles, and all of these kind of other properties to separate the cohort and identify how this particular gene then associates with it. So if you have this data set, you have differential expression analysis, then another thing that you can do is you can use this, of course, as a table, as we've been using thus far, but you can also make this look differently. So the type of display that we have for this result can be the list that I've shown you before, but you can also convert this into a graphical representation, like, for example, a volcano plot. So by just switching this lever, you now represent the same data that we just saw, but now in the way of a volcano plot, where you have the log fold change between the different groups on the x-axis, and on the y-axis there's the log of the p-value. And then, of course, meaning that the farther outwards you are, the higher the fold change is, the higher the points are, the better the p-value is. So these are also interactive in the platform. So essentially what you can do is you can use, again, this gearbox over here to make all kinds of adaptations to this plot. So a thing that you can do is you can just use your left mouse button and click on any of these elements, and then it will automatically annotate the gene that is, in this case, indicated. That can be changed in size, color, properties if you wish, by using these elements that are over here. And another thing that you can do is you can also indicate, for example, where your significance thresholds are, which are the genes that are significantly differently expressed, and then you can just color these dots on the basis of that. All of these parameters are something that you can also set. What you can also do in addition is you can use quite a large signature database that we already have in the platform, and where you can also add your own results to. For example, indicating that you would like to see kinases highlighted, or transcription factors, or whatever is already possible in the platform, and then you can just simply add those as color elements as well. Oh. And then finally, the thing that you can do over here as well is if you use the right mouse button on it, essentially you will go back to this detail view where you can see what this p-value and fold change looks like if you really look into the nasty details of all of the samples and how they are segregated. And from there, of course, you can perform the rest of your analysis as well. Another type of visualization that you can use with your results is using these group change bars plots, which essentially indicates the fold change by a bar plot. And also here it is interactive in the sense that if you click on it, it will show you again the underlying data for this particular test result. And that's also kind of the thing that I would like to bring across today from the perspective of the platform, is you can do a lot of different analysis, but it also allows you to go a couple of steps deeper into your data to try and understand what's going on, or maybe see something that is peculiar, looks a little bit off, and where maybe you're looking at something that is an artifact, but maybe also your next Nature paper that you may identify by just looking into something that looks a bit off. So once you have your result set, of course you can look into the detail of every single gene. It's going to take a lot of time and your attention span will not most likely survive this. So there are also a lot of analysis that you actually can do with the result itself. So first of all, you can constantly adapt the kind of properties that you would like to take into account. So in this case, I will now just reduce the data set to only use the top 1000 results by just using this lever. And then I will, for example, start doing a gene set analysis. A gene set analysis allows you to look at overrepresentation of your genes that are differentially expressed versus a collection of signatures that you want to use. And this collection of signatures, we already have over 100,000 of these signatures preloaded in the platform, but you can also add your own if you are interested and start using the platform and store your own results. So what you can then do is you select one of the resources. In this case, I'm going to use the broad molecular signature database, hallmark signatures. And then essentially you can just run this gene set analysis, which looks for overrepresentation. Well, as you already could have seen in the volcano plot that I just showed you a couple of minutes ago, it was really distorted towards the right-hand side, so most of the genes are now upregulated in this particular example, which is a little bit unfortunate because here also then everything will have one color. If it's a bit more harmonized in that sense, you will also see that the genes that are popping up here as significant and interesting results can have like a red color and a green color, or if you use multiple groups, another color, so that you can immediately see that there may be a confounder aspect on the signature that you're finding, and that you can then take into account running the next step of these analysis. So there is also this indication here of this H, which is called, which will link you to a heat map. So then you will use this particular signature that was identified. So in this case, we picked up epithelial to mesenchymal transition. Of course, we used CMS422, right? So this is also kind of what we expect. It's just to demonstrate how these kind of things work. And then if you click on that one, it will show in the platform a heat map where you can then see how all of the different genes within the signature are actually behaving. And in this particular signature, you can see that most of the genes that are in here kind of do the same thing, right? They are very much correlated because you see these vertical bands that are occurring. And also here, what you can then do is you can use a grouping variable, for example, or filters to then adapt this visualization in such a way that you can now see, hey, how does this now relate to this different CMS subtype that we started off with. You can also reduce the data set again to only show CMS2 versus 4 if you want to, and then all the heat map will just constantly adapt to that. So in that way you can get a bit of a deeper understanding of the signature that you picked up and how reliable the signature that you're picking up actually may turn out to be, as is the case over here. So another kind of analysis that you can perform, I'm not sure if it's biologically the most sensible one, but it's just showing you the principle that I would like to produce here as well. So we can use these 1000 genes that we picked up in the differential expression analysis and then ask the question, can we use those 1000 genes to take our cohort and segregate it in such a way that we can define X number of groups? And this is called a k-means analysis. And what you actually can do with the k-means analysis, we can just use this button that we have in the platform, and then here you can set up the analysis as you see fit. So we're going to take these 1000 genes, we're going to ask for separating this cohort in 2 different groups. Is this possible? Can we do that with these 1000 genes? And in order to get some significance and statistical value out of this, we can use a number of repetitions on running a k-means analysis. So essentially what R2 will do is it will run the k-means analysis 10 times, picking the best separation out of 10, and in that way then demonstrate or show to you how easy it is to separate this cohort on the basis of those 1000 genes. And essentially what happened here is that it actually did this particular analysis, and what you can see on the top here is again a heat map, where this is the track of the CMS classification that we had, and then the thick one over here, if you would zoom in, it's actually 10 times running this k-means analysis in a row. And if it looks like this, completely separated in these two different colors, it means that every time it did it, it found the same solution, which means that it probably is quite a strong effect. If you start looking into things that are a bit less easy to separate, you will find that these color codes are completely running through each other, and that will immediately spark your interest that maybe it's a bit more difficult to do it in this particular way. And then out of all of those runs, it will take the majority vote to essentially attempt to provide you with a new grouping variable that actually then was the basis of this k-means analysis. And that particular group has then also been tested immediately for you against all of the annotations that were already in the data set. So you can immediately see, hey, the separation that we made seems to separate how many variants are in a particular sample, or whether it has lung metastasis, etc., etc. All of these kind of things are immediately tested, so that you can also see if this associates with something that you already knew, and whether that makes sense or not. Yet another way in which you can run a next analysis on top of this differential expression is using the thing that we call the chromosome map. And a chromosome map does exactly as it tries to tell you again by its name. It will associate the differentially expressed genes that we had on chromosomal location. So is there a particular region in the genome that appears to be over or underrepresented if we look at your differential expression analysis? In this case, a couple of chromosome arms are being picked up. So, for example, chromosome 19. So you can take then that result to our own embedded genome browser that we have in R2 as well, which will then allow you to show the results again, but then in a bit more detail on the chromosomal location, where you then can identify where are these genes located, are they primarily upregulated or downregulated, that you can see depending on the color, and from there on you can also get back into the platform again and look at it in detail. And I think one of the last things that I want to show you on the differential expression analysis is you can also fork out to tools that are available on the internet as well. One of them being really quite frequently used is called Enricher. Enricher also allows you to do kind of like a gene ontology search or gene set analysis, where within R2 you can set it up to execute in a particular way. So here we're now going to use only those genes that are more highly expressed in CMS4 than in CMS2. And then you simply can just submit it. It goes to the Enricher website and immediately runs the analysis for you, such as you can see pathways, ontologies, all of these kind of signature databases that are automatically tested on your dataset. And as the way in which we are running this analysis here is that we also provide a background file with it, such that Enricher knows what has been checked and what is significantly different, which means that it automatically also corrects for impossible combinations that may show very high relevance. Let's say you only take kinases for your search, and then you take that to the Enricher database. If you don't put a background to it, it will say, hey, I find kinases, which is not really something that is of interest if you only started with kinases, right? So then it takes this into account so that it knows what kind of elements it should focus at. And then another thing that I always suggest people to use is that if you run something, you find something of interest and you want to keep track of that, you can also save these results as text files. And those text files can be pretty easy if you understand how they are put together. It essentially just gives you back the result that you got from your differential expression analysis with the reporters, in this case ensemble genes, gene names, the p-value that's associated with it, what kind of grouping is in it. But what I think is also very important, it also provides you with everything that you would have to set in the platform to reproduce this analysis again. And there are certain functions in R2 where you can actually feed back this text file, and then R2 will run it again, and you can actually progress on top of that results. Another way in which you can start working with your data is to store the data within R2 itself. So differential expression analysis gives you a list of genes. Those genes can also be put back into the platform as your own signatures. Right? So then you can say, these were the genes that were differentially expressed between CMS 4 and 2. And later on, I may want to use that as a exclusion criteria or an inclusion criteria to only use those genes. Or later on, maybe identify that something that I picked up now appears to become a result if I run another analysis and do a gene set analysis. So all of these things are possible by just saving them in your own account. And these are then personal tracks that are personal gene sets that actually are within your account. So thus far we've looked at the list, we've zoomed into particular genes. You can also make visualizations where you can look at more than one gene at the same time. So this is view the multiple gene option. And here what essentially you can do is you can provide a little list of genes that you would like to have a look at. And then you can represent them in different ways in the platform. And in that way you can build visualizations that allow you to look at a number of genes in the same at the same time, which can either be the complete data set or segregated by a track, colored by another track. So you can make infinite combinations also here in terms of how you would like to visualize these things. So another type of analysis that you can perform within the platform is actually where R2 got its name from. So this is how we actually got started, and that is correlation. So R2 stands for the correlation coefficient. And this has been at the birth of our platform. So there are a lot of things that you can also do running these types of analysis. And here I'm going to show you one example using a neuroblastoma dataset. So in this case we're going to use a neuroblastoma dataset of 88 patients. This is from an older microarray platform, which is also something that I think is pretty cool from within R2. You can run RNA sequencing, you can run microarrays, you can even do SAGE, all of these like ancient technologies, and you can use the results that you obtain from one to validate the other. So here we're going to look at correlations. Like I already said in the beginning, MYC is a gene that's important for neuroblastoma. If you have a lot of copies, that's a bad sign for the patient survival. And what you can also show within the platform, if you have multi-omics data, which we have for this particular data set, is that you can assess the DNA versus the RNA expression as well. And here you can then clearly see that if you have more copies DNA of MYC, so these are the amplified samples, you can also see that the expression of MYC goes up. So we could then wonder, are there other genes that show a similar kind of pattern as MYC, right? So can you also find other genes that may co-amplify or co-express or co-upregulate their expression? So then you can run this correlation analysis. And what I'm going to do here is I'm going to use not all of the genes to scan, but an intersection out of this. So we're going to use this gene set database that we have. And there, for example, we can identify only genes that are participating in the cell cycle. So once we've made that distinction, we can then run the analysis, which then is going to be really fast because it's only a couple of hundred genes. And now what we can see is we can find out which genes that are associated with cell cycle show an association with the MYC expression. And then you pick up all kinds of interesting candidates. Again, you can click on it. In this time, the visualization here is a YY plot. So it's essentially the same thing as looking at one gene, but then at two in a row, showing you sorted on the expression of one gene, in this case MCM2, what the expression is of MYC-N. And again, you can use these color codes underneath to see if there are particular associations that you find of interest. So once you identify a significant analysis, so this has a p-value of 10 to the minus 6, so it should be quite an interesting one. You can then also wonder, did somebody else already publish on this? Has this been identified by somebody else before? For that, we have the tool called PubSniffer. And what PubSniffer does is it will go to PubMed with a live query asking, do you have any abstracts that mention both MIC-N and MCM2? And then it will tell you, well, I find one of those. It will also use both of your genes in isolation and identify how many papers are available for MIC-N or MCM2. And depending on the data set, we also can put in keywords. And every keyword will also be associated. So this is a neuroblastoma data set. So we've also included here neuroblastoma. If you have a colorectal cancer data set, it will associate colon with it or something like that. This is all something that you can also instruct. And then once it identifies something with both of your genes in it, it will also check the abstract itself if maybe there's a sentence where both of the genes are being mentioned. And if that's the case, it will plot it underneath. So in this case, we know that there's even one publication that has mentioned MIC-N bound to the promoters of MCM2. For the sake of what I'm going to show you here now, we're going to forget about this, that this is something that's known. It actually came from our department. This is also why I like this example. Oh, one thing I forgot about is, there's another tool that we also have, PubReminer. PubReminer is a frequency table analysis. So essentially what you can do from there is you can take a particular query, any query that you put into PubMed, and then it will get all the abstracts out of it, shred it into words, and then you show everything as frequency tables. So you can find prominent authors, times when literature was accruing about something, what kind of keywords has been used, and you can actually click on all of these elements to make the query smaller and smaller and smaller, using information that's actually based from within these abstracts, and thereby making a smaller selection of something that you may want to read. Also pretty nice program. So if you have a correlation analysis, the way in which most people like to visualize that is in an XY plot, right? Because then you can more easily see whether it forms kind of a cigar. So here you can see that there is a nice trend in there. 10 to the minus 6 is the significance. You can then also wonder, so if you have a relationship between two genes, it can either be that one gene is regulating the other gene, right? So upregulation of one upregulates the other, or it can be that both of them are maybe regulated by an upstream process. They are just both the target of a particular association between them. And in order to actually help people identify such properties, we have a couple of different tools that we have implemented that may help identify something interesting. One really needs very specific data, which is the example that I'm going to give you here. We have a lot of time series experiments in the R2 platform as well, which means that basically somebody tried to mess around with a particular gene, followed it in time, and look and identify what happened to it. This is a specific part of the R2 platform where you actually can look into these time series experiments. I'm not going to dwell on them today, but what you can do is you can use all of the things that are already implemented in the database to ask the question, I had MIC-N, I had MCM2, do you find something of interest here? And then there are a number of experiments that have been performed where essentially from there you can pick up something that can be very helpful in explaining things. So here we have a neuroblastoma cell line where NMIC has been downregulated by a lentivirus, and what you actually can see is if you run this experiment, MCM2 drops, which is a sign that maybe the relationship between those two genes is a direct one. Another way, we have an experiment here, another neuroblastoma cell line where NMIC was introduced. It was actually added to the experiment. And what you can then see, MCM2 going up. So both ways, it appears that if you mess around with NMIC, then something happens to MCM2, pointing to a direct relationship between the two. And then we also, and I will come back to that completely at the end, we also have chip sequencing data in R2, quite a number of profiles for it. So what we can then ask is there maybe binding of NMIC, which is transcription factor, on the promoter of MCM2? And then also here, you can find a number of different experiments where essentially you can see that this binding is happening. And this has been the basis of this particular paper that we just had a look at. So you actually can use different options in the platform to test part of your hypothesis and then maybe continue on it. Admittedly, not too many examples are available to this depth because, of course, some data has actually been created to generate this example. But what you can use is the fact that we have these thousands of different experiments already loaded in the database. So if you have a relationship between two genes, we can look at the 2D distribution option that we have in R2. And essentially what that will do for you is it will take the concept of these two different genes, MCM2 and then MYC. We know that in our data set we had a significant, nice positive correlation. So it will now just ask in hundreds of other data sets, what's the relationship between MYC and MCM2? And then it will show this as an average expression of your one gene and MYC, average expression of MCM2, and then the color of the elements will show you the correlation. So you essentially can identify other data sets where also this relationship has been found, which will then maybe instruct you that, hey, this is found everywhere, pretty interesting, or it only appears to happen in this particular other data set, and then maybe you can have a look at that and see if this may help explain what you're seeing in yours. So also these are hyperlinked. So if you click on it, you will now see another data set here, a retinoblastoma data set, where essentially you can also see this very nice positive relationship between MYC and MCM2, and that may be helpful in trying to explain what's going on there as well. Another way in which you can actually explore your data deeper and further is to make just use of the fact that you have this correlation plot. We have a database with all kinds of annotation features in it. So what you actually can do is if you use these extra tabs that you have in the toolbox, you can switch on this thing over here, which is called the linear fit. So that's actually trying to fit a linear curve between your data points. And then essentially you can see this is where the R-square value is actually being calculated from. You can see the relationship between your data points and this line that can be defined. What you can then also do is you can use the fact that you can color all of these elements by an annotation feature. So in this case what I did is I'm going to color them by MYC and amplification status yes or no. And by doing that, R2 immediately now also changes this linear fit into two different linear fits based on the different subgroups. And if you get the concept of that, that also gives you a very powerful option, because essentially I could do this for every different feature and then maybe identify something where I see the correlation is particularly high in a subgroup but not in the others, or the other way around. And for that we essentially have a button as well implemented in the platform. We can just say find all of these associations, and then it will just test everything, every subgroup for you in the platform, thereby then identifying, hey, if you use age in a particular group, there the association is really strong, or whatever you find of interest. And also there you can click on these guys and then it will show you the picture with that track selected, such that maybe that can help you identify a relationship that only appears to apply if you are in a subgroup of your disease. And as you will maybe later on also discover, there are many of these little loops in the platform which allows you to perform such an analysis. If you ever get the idea that you want to do it, then often there's already a button for it that allows you to do it. And if you think of something cool that's not in there yet, please let us know, because this whole program has developed by getting feedback from the people that have used it. We are using it ourselves as well a lot, but especially all the feedback that we got from users helps to make things better. So this was the first getting started of what I wanted to show you here today. So what we're going to do now is continue a little bit with some more extensive examples, and then I'm going to end with a few overview slides of all the kind of things that are possible.